Glycolysis. Glycolysis is basically the breakdown of glucose. So lysis, right here means so in this part, lysis means breakdown, and glyco means glucose. So what happened exactly and when does it happen? So glycolysis happens. So if we're having, this is our bloodstream. So this is in the bloodstream. So when the glucose levels are really high in the bloodstream, what happens? It keeps going inside of the cell in the cytoplasm, and the concentration will be high inside of the cytoplasm. So the first thing that happens, glycolysis will occur, meaning that right now we're having too much of glucose, so we need to turn that glucose into energy instead of just having it all around like that. So the first step that occur is attaching a phosphate group to the sixth position of glucose. And the main reason for doing this is to prevent the glucose going back there and getting out. So right now it cannot go out. If glucose wasn't phosphorylated, it will be able to go and exit the cytoplasm again and go back to the bloodstream, and that will cause a lot of problem after that. So this is the first step in glycolysis. So they want to trap the glucose from going out back there. So the first thing is the phosphorylation of glucose. So right now we're having glucose being turned into glucose 6-phosphate. And the enzyme responsible for this, phosphate, the enzyme responsible for this is either hexokinase or glucokinase. So what is the difference between them? Hexokinase is in all of the body cells. In glucokinase, it's in a little bit more specialized cells. We have it in the liver cells, and we have it in the beta cells in the pancreas. So that would be our first step. And in this step, it's irreversible. So whenever this happens, it cannot go back again. Okay, it cannot go back again. It's just in one direction. And it uses ATP. Okay, turn it into ADP. And as you can see here, there is triphosphate, and it turned it to diphosphate, meaning that the enzyme had used one phosphate to put on the 6th position of glucose. Now we're going to the second step here, turning the glucose 6-phosphate into fructose. Fructose 6-phosphate. And in this step, we're using phosphoglucose isomerase. Phosphoglucose isomerase. So from the name of it, phosphoglucose isomerase. So isomerase is basically it's just the turning of the component itself, despite the fact it has the same structure. So glucose and fructose, it's a 6 carbon molecule. So it just changes a little bit to turn it to fructose. And then after that, right now we're gonna have another irreversible step here, which is phosphofructokinase 1. So this step is also irreversible, and it has the product of 1,6 fructose. I mean 1 fructose 1,6 biphosphate. And then also, as I said, this is a irreversible step. So it also uses ATP, so it uses energy and turns it into ADP. Also, as you can see here, it was only 1 phosphate on the 6th carbon of fructose. Right now it has 1 and 6, so it has two phosphate group. And whenever you see that there is a turn from ATP to ADP using one of the phosphates of the ATP or using the energy, you're going to know that this step is irreversible. We have 3 of them. These already are 2 of them. The third one is going to come up. Coming up to this is aldolase, which is the enzyme that will convert this fructose 1,6 biphosphate into 2 3 carbon molecules. So aldolase, and it will be turned into 2 molecules, as I said. Each one of them is 3 carbon molecule. So we're having cholesteroldehyde 3-phosphate. 3-phosphate. And as I said, it's the enzyme responsible for this is aldolase, and the other product will be dihydroxyacetone. And even this product will be turned into cholesteroldehyde 3-phosphate by the action of triose phosphate isomerase. So after that, dihydroxyacetone will be also cholesteroldehyde 3-phosphate, meaning that we're going to have 2 cycles right now. So we're going to have this cycle going in and this cycle going in. So both of them are going to have the same purpose, and the purpose is getting to the product of pyruvate. So I'm going to go with one of the cycles. So that means if we're having, let's say, 2 or 3 ATPs in one cycle, it has to be multiplied by 2 to get the whole process, what is the net process of the ATP. So so far, we have used 2 ATPs. So right now, we're at -2 of ATPs. So let's see what's going to happen here. For cholesteroldehyde 3-phosphate, the first thing will happen, this product will be turned into 1,3-bisphosphoglycerate. So, and the enzyme responsible for this will be glyceraldehyde 3-phosphate dehydrogenase. So whenever you see dehydrogenase, you're gonna know that NAD molecule will be there. So NAD will be used and will be reduced to NADH, and NADH is a very high energy molecule, so it could actually yield for 3 ATP. So 1 NADH will yield for 3 ATP. Okay, and then we're having after this the 1,3-bisphosphoglycerate will be turned into 3-phosphoglycerate. And the enzyme for this would be the kinase. So it would be, in this case, phosphoglycerate kinase. Okay, kinase. And in this process, ATP will be actually produced. So ADP would be taking one of the phosphate groups here. So we'll take the one in the first carbon and will be added to the ATP. Okay, so right now we're having one ATP here, just produced ATP. Okay, so and then 3-phosphoglycerate will be 2-phosphoglycerate using the enzyme phosphoglycerate mutase in this case. So it'll be phosphoglycerate mutase. Okay, and then after that, using the 2-phosphoglycerate and converting it into phosphoenol pyruvate. So now we're so near to the pyruvate, and the enzyme responsible for this change is enolase. Okay, and then lastly, we're gonna have pyruvate kinase as the enzyme, and from the name of it, it will be dephosphorylation. So this phospho group here will be taken, and will leave us with pyruvate. So ADP would be turned into ATP. Okay, so there's energy that will be there. So this is another 1 ATP here. Okay, and our pyruvate at the end will be ready. Okay, so if we're going to count, this is glycolysis, and it happens in the cytoplasm, as you can see here. This is the cytoplasm. So after that, the pyruvate will actually go into the mitochondria, which is, this is the mitochondria. The pyruvate will go in there, and the citric acid cycle will occur there. But we're not going to talk about it in this video. So if we're going to count how many ATPs and how many energy molecules are there, so as I mentioned, there were 2 ATPs that was used at the beginning of glycolysis, right? So, and this is -2. So right now, what is the net of the ATPs that we got here? So we got one here, 1 ATP here, and we got another 1 ATP here. So this is another one, 1, 2, and we got 1 NADH, DH. Okay, so if we're gonna do the math, as I said, this is 2 cycles, as you can see here. So there is this cycle too that will have the same exact products. So we're gonna multiply this by 2. So this is 1, this is 2 ATPs, and here's 1 NADH. So we're multiplying every single thing by 2. So 2 NADH, and we're having 2 ATP. And as I said, NADH is actually this is how to get energy through oxidative phosphorylation. And for the ATP, this is the process called substrate level phosphorylation. So this is how to get energy through ATP, phosphorylation. So right now we're having 2 ATPs, we're having 2 NADH. And as I said, NADH, 1 NADH would yield for 3 ATPs. So right now we're having 2 times 3, and this would leave us with 6. And here's the same thing, 2 times 2, because we're having 2 cycles, will be 4 ATPs, okay? So 6 plus 4 will give us 10, right? And as I mentioned, there are 2 ATPs that were used at the beginning of glycolysis, so we're gonna subtract that from 10, and the net of glycolysis would be 8. Now there is a very important thing. So pyruvate, the fate of pyruvate would be determined by if there is oxygen or no. So there would be aerobic condition and anaerobic condition. Okay, so in this case, for pyruvate, there will be it will be able to enter the mitochondria, and in this case, yeah, there will be 8 ATPs that will be synthesized. But in the anaerobic, it's not going to be the same thing. Despite the fact pyruvate is not going to be used either, but the net will be only 2 ATPs, so it won't be 8 ATPs as in the aerobic. And the reason for this is because in the anaerobic condition, NADH is not going to be used. NADH is not going to be used as an energy source. It will be used as an agent for turning pyruvate to lactate. So let's see here. As I mentioned before, so we have said that glucose is turned into what? Glyceraldehyde 3-phosphate, right? 3-phosphate, right? And then from there, this glyceraldehyde 3-phosphate would be it would go through dehydrogenation, 1,3-bisphosphate. Phosphoglycerate, right? And I said dehydrogenation means that it uses NAD, okay, and the product was NADH was produced, right? So, and from there, 1,3-biphosphoglycerate would be going to be pyruvate, right? But then pyruvate, due to the lack of oxygen, it won't be able to get into the mitochondria. So what's going to happen is that the NADH that was formed before, it will be actually used as a molecule, as a reducing agent to reduce pyruvate into lactate. Okay, so right now NADH will be turned again into NAD, which is so essential for glycolysis. So this step is actually mandatory. So if there is no oxygen there, then the NADH has to be turned into NAD. Pyruvate has to be turned into lactate. Okay, this is in the absence of oxygen. So right now, this is the reason why in the anaerobic condition there is only 2 ATP, and there's no NADH as an energy molecule, because NADH is not going to be used as an energy molecule in this case. It will just be used as a reducing agent, giving the H to and it will get lactate at the end as a product. So in aerobic condition, 8 ATPs will be produced. In the anaerobic condition, it will just have 2 ATPs. In aerobic, it was able to use the 2 NADH to yield 6 ATPs. As I mentioned, 1 NADH would yield for 3 ATPs, while in the anaerobic, it used the NADH as an agent to produce lactate. Okay.